Hello, everyone. I am Flavia Katerica. I'm the co-organizer with Dana of this symposium. And so I'm very excited to see all of you here today. And I have the duty of and the pleasure of introducing the second act. The second act that we've seen in the first act, the history of malaria from the very past to the present. And now in this second part of the program, we will focus instead on the challenges. And I realize that there is no break, which it might be a little bit tough for people. So if you want to stand up for a second and stretch a little bit, feel free to do that now. Yes, exactly, because it might not be possible for the next hour. And so the challenges that we will discuss now are challenges that are caused by humans, by people, by us, like climate change, although it's very controversial to say that it's man-made, but yeah, maybe I'll say that. And then also other environmental challenges that will be discussed, as well as these challenges due to human migration that is forced by conflicts, other humanitarian crises. And then we will end with challenges caused by the failure of our therapeutics as well as by our diagnostic tools. And so with that, I'm happy to introduce the first speaker, who's Professor Marcia Castro, who's an Andalou Professor of Demography and the Chair of Global Health and Population Department at the Harvard Chan School of Public Health.
Thank you. All right, so second act is going to start with taking you all to the Amazon. So I'm going to talk about the challenge of different types of environmental change, but also on climate. And I'm going to use the Amazon, the Brazilian Amazon specifically as an example, because that's where most of my work is. I hope I'm going to convince you that in this context, which is very similar also for other countries that share portions of the Amazon, Peru, Colombia, is that those things are related. So I'll show you that this environmental change in this particular context is actually speeding up this process of climate change. So those are not completely separate things. All right, so what is the idea here? Just to give you some context, that's malaria in Brazil from 2003 to 25. The line shows you the total number of cases, so there's sort of a recent decline, not so fast, but after it reached almost more than 600,000 cases. And the different colors show you the percentage across the total number of cases that happen in rural areas, urban settlement areas, indigenous, and mining. And what we see here is that in the beginning of the time series in 2003, we had less than 3% of the cases happening in indigenous areas, less than 5% in mining. Then 2019 was the first time ever that more than 20% of cases happen in indigenous areas. Following year, first time ever, mining was 2 digits in percentage points, so more than 10% of all the cases. And since 2023, when you put the two together, cases in mining and indigenous areas, that's more than half of all the cases in the country, and it persists. So it was the same in 24 and 25. So there is a shift when you look at the composition. If you only look at the curve, everything looks fine, although the decline is not fast. If you look at the composition, there is a huge problem that we need to look at. So first reason behind this problem, deforestation, mostly illegal. And if you look at this map in the middle, everything red is where the land has been cleared, where the forest has been removed. It may sound funny when you see those areas, right? Those are the indigenous areas. They are buffers for the deforestation. And if you look at this graph, that shows you the deforestation per year since 1988. And what we see is massive programs that were done that really brought deforestation down. That was the first 2 years of the government of Lula, who is, again, the current president. That's what happened under the previous government that relaxed environmental laws in Brazil. So deforestation was going up like crazy, and now it's going down under the current administration. And last year, 2025, it recorded the third lowest volume of deforestation in the Amazon in history. So as you can see, Brazil has been really good in bringing it down, failing dramatically in keeping it going down. So it comes up, and this pattern has many implications, not just for the environment, but also for health. When we put malaria into this picture, this map has two variables, both deforestation and malaria. Everything that is this dark color here is where you have both high deforestation and high malaria. As you can see, it's happening in what we call the new frontier of deforestation, not the old one that we call the arch of deforestation. So there's also a temporal component in how we understand removing the forest cover and the increase in malaria. So there's a lot of work that has been done trying to basically quantify how much that matters for malaria. Using year, that's the wrong temporal scale. Malaria has seasonal patterns, has a temporal component, as I showed you. We were able to do this by month. And then what we showed is that every 1% increase in monthly deforestation leads to, on average, 6.3% increase in malaria. But there is variability across the Amazon, as you can imagine. The area is 60% of Brazil. Brazil is quite big, in case you didn't know. So 6.3% is just an average. It varies a lot. Interesting is the more greener the surrounding area where you're having the deforestation, the highest the impact. And again, that has to do with this temporal component of how deforestation affects malaria. So that's one. Two, mining. That's another type of environmental change. So there are two types of mining we can look at. Everything here in red and in the map in red is industrial. That's not what we're worried in the Amazon. In the Amazon, we're worried with the yellow ones. That's the they call artisanal mining. There's not much artisanal about it because there's heavy machines in there. But it also has expanded dramatically in their, I'm probably in front of it, but under this recent government where we had more of this type of mining, it's all gold, than the industrial one. That's a typical area of mining. This is in the Yanomami indigenous land. That's an airstrip, illegal. That's the mining, illegal. They use mercury, so malaria is just one thing that happens for health. There are many other negative aspects. And the villages where the indigenous population are very close. Most of this mining is illegal. And if you take a time series from 2018, from 1985 to 2022, almost 41% of all the mining open in the Amazon happened in a period of only those years, 18 to 22, and almost 63% of all the mining in indigenous lands also were open in that short period of time. So again, fast, devastating, and was intensified by the kind of leadership Brazil had during those years. What's the consequence of that? I'm gonna give you an example using the Yanomami indigenous land. That's this area here, the largest indigenous area in Brazil. It's about the size of Portugal. Each dot in this map is an airstrip, like the one I showed you in the picture before. Any one of those dots on top of a green area, protected lands, or blue area, protected indigenous forest should not be there. And if you take the Yanomami with this recent expansion, this green line is the expansion of garimpo, of mining in the Yanomami indigenous area. 1/5 of all the villages, of all the different communities of indigenous people in the Yanomami were within 1 kilometer from an airstrip. So you can imagine all the devastating effects for those communities. That shows you how much the two maps, how much malaria increased in the Yanomami area. The green areas are basically a buffer of 5 kilometers in the mining area, so it's more visible. And what is interesting to note is that you had an increase even in areas that don't have the garimpo. So what's going on? It's the mobility, and Dan is going to talk more about this. There were different routes the miners were taking to go to the capital for treatment or for whatever they needed to bring back to the mining area. And some of those routes are exactly those areas where you also had increase. They're carrying the parasites, the mosquitoes are there. So, the increase in transmission goes way beyond the area where you have the garimpo. Again, just on average, 1% increase in the mining area in the Yanomami was associated with a 24% increase in monthly malaria cases. And we did a simulated scenario, basically trying to answer the question, what if the mining had not increased exponentially as we observed, starting in 2018, and that's more than 102,000 cases that could have been avoided. Okay, regarding climate, I think, first of all, what we need to understand is that there are direct and indirect effects of climate. The direct basically deal with the geographical expansion of where you see transmission and the intensity. That's basically what it is. You can have malaria in places where you didn't have before. You can have, in some places, it recedes, and in some places, the transmission can change or nothing will dramatically change. But the indirect effects are also important to consider. Some of this is related to the health systems. If the health system is not resilient to climate, facilities can be destroyed. That can affect the workforce. It can change the coverage of services, the quality of services, how much the cost that you have to put in to do the malaria control. Some communities can be completely isolated, for example, depending on the extreme event you have. So all of this has to be considered, and it's very complex. Second thing you need to take into account is when you are dealing with the tropical forest, the process of environmental change, and the two that I mentioned, deforestation and mining, they are speeding up this process of climate change. So the southern portion of the Amazon, for example, is already receiving less rain because of the removal of the forest cover there in that arch of deforestation that I mentioned before. And the reason is not only the trees produce rainfall, but in this particular ecosystem, the Amazon is absolutely critical for the hydrology and climate of the entire region because of the rains that it brings to the southern portion of South America. So again, those two things are directly connected. 2023 and 2024, the Amazon region saw the worst drought ever recorded, both in intensity, but also given the fact that it started much earlier than usually is observed. You can see in those two maps, everything that is this dark red is exceptional drought. And again, it affected not just in Brazil, but the Amazon biome extending to other countries. So you can understand, for those of you that have never been in the Amazon, this is 23. This is the same place. You can see those houses here, those houses here. The rivers basically dried out. I mean, there's some rivers that look like you're in the ocean. In case you've never been there, you only see water, and they became like those little streams. This is the Negro River. It's the largest black water river in the world, and it became completely unrecognizable. This is one area in Manaus, and again, you can see the big river just became this tiny thing with water of extremely poor quality. If that was bad, 24 was worse. This is one of the main ports in Manaus where a lot of food comes in and people take boats to go to other places. And it was very restricted on the kinds of things that could get in here. Several communities became isolated for months. no kind of service could go there. And that's one typical house that basically are always floating in the rivers, and 2 minutes, okay. So what we did is we looked for Brazil, we looked at those data, and Brazil has a lot of data. That's one of the cool things about doing this kind of work. This basically shows those maps here in the bottom. This is La Nina, this one is El Nino. By trimester, we're able to see what is the change in malaria transmission during those events. Overall, if you just look for the Amazon, you tend to have fewer cases of malaria in both of those events. But when you look at those maps, you can see that some areas are going to be pressured because they're going to have more, while others have less. So if you just do a study for the entire Amazon, I'm probably not going to pay attention at all because it's meaningless. And that's the importance of scale, to be able to inform action that can minimize the transmission. And that's a clear example. So there's also, I'm just talking about the Amazon. So I want to show you there are other studies that are starting to look more closely at this impact of climate. That's a work that was done by Boston Consulting Group and the malaria map. Again, it's country level for Africa. Same thing I said for the Amazon, but it's a start to show what could be the likely impact. Those are scenarios, but they're important. So just to conclude, just as we have many challenges, there are also opportunities to be creative on addressing those challenges. Those are some of the things that I've been doing with the National Malaria Control Program and in local governments in Brazil. And we already talked a lot about new technology. And Dyann, you forgot stafenoquine. How could you do this? Right? Anyway, it's a new drug. So there is a lot that can be done, but there's also a lot going to the basics and doing the best you can with the resources you have, including work with people on the front lines dealing with control. We should not forget this. If you don't do this, you can have the most outstanding, phenomenal technology. If those people are not on board, it's going to fail. So with that, I just need to show you the Amazon. Thank you.
Thanks so much, Marcia. Very dramatic images of the Amazon. And the next speaker is my co-host, Dyann Wirth, who is Associate Professor of Immunology and Infectious Diseases at the Harvard Chan School of Public Health. And he's also Associate Director of the Genomic Center of Infectious Diseases at the Broad Institute. So Dan, welcome on stage.
Thank you. All right, so we're going to stay in the Americas for a little bit. Marcia was talking about climate change and severe weather. These kinds of things are a driver of migration, along with economic and political factors, and migration has important consequences for controlling malaria. So as we were discussing, there has been progress against malaria in the Americas, in part because it is a lower burden part of the world. So the most recent country to be certified as malaria-free by the WHO is Suriname, a much smaller country than Brazil, but it wasn't easy there. 22 decades ago, they had 80 to 90,000 cases a year in a population that is smaller than Boston, and it took a lot of effort with improved diagnosis and treatment in order to achieve elimination. But this elimination in Suriname, it's a fantastic achievement, but it's fragile in part because of heterogeneity and progress against malaria in the region of South America, in particular Venezuela, which has had a rough time in the last decade with cases surging from about 40,000 a year to 450,000 a year a few years ago. And because of economic drivers like artisanal gold mining that does lead to crossing of borders, it's very difficult to think about elimination in a single country context. And in particular, the question has arisen as to whether human migration has contributed to a recent increase in cases in the last few years in Mesoamerica and several Mesoamerican countries. So the number of migrants crossing from South America into Central America on the way northwards to the United States through the Darien Gap region of Colombia, right here, where it joins of Panama, rather, right where it joins South America, has spiked in recent years, as indicated by this gray shaded area. And in tandem with that, apparently, the increase in the number of Plasmodium vivax cases in Panama, Honduras, and Guatemala has also increased. And so one question is this directly attributable to migration, and can this inform how those countries address rising cases? And so we heard from Megan and Manoj about getting malaria DNA out of gorilla poop or out of fossils. That's pretty hard, both of those tasks. You can actually get DNA out of blood or dried blood spots on paper much more easily and cheaply. So that's what we do to try to understand things. So I just wonder, how many people in this room have spit in a tube and put it in the mail and sent it off to a direct-to-consumer testing service? I've done it, so don't be shy. Yeah, quite a few hands. Yeah, so tens of millions of people have done this. And as a result, these companies have all of our private data. And then they can tell you lots of things about yourself by reading your DNA. So this is actually my ancestry profile. And a lot of this I knew, but in fact, my mother was adopted, and I didn't know about my ancestry from that side of the family. And I discovered that I likely had a grandfather, or perhaps a couple of great-grandparents who came from Eastern Europe, likely from Russia or Ukraine. And by this information, because I'm in North America, we can infer that one of my ancestors made the trip from Russia or Ukraine to North America at some point in the last few generations. And so we can do a similar thing with malaria parasites. They do not use the same companies, but we can sequence their DNA from various places, and then we can reconstruct the degree of connectivity between those places based on these familial connections between parasites that we can measure in the DNA sequences. And so without a company handling this task, my group has been working on building a better database of the genetic profiles of malaria parasites from lots of countries in the Americas, in part to better understand the connectivity of parasite populations and understand the role of migration in sustaining transmission. And so we've sequenced around 6,000 falciparum and vivax malaria samples. Contrary to what Manoj suggests, I like both parasite species equally. And about 4,000 of those genomes have turned out to be really high quality and useful for understanding connections between places. And so this is a plot made by Pablo Manrique, a student in my group. He's not a student, he just defended his thesis last week. So Dr. Manrique Valverde created this plot on the left, where each dot is a parasite sample, a sample from a vivax malaria infected person. The colors correspond to the countries they come from, and the lines between them indicate that they're family members. They share recent genetic ancestry. And you don't have to see the details of this plot, but you can see lots of individual speckles of singleton parasites where we haven't actually captured any recent relatives in this sample. And then you can see these balls where they're all connected in a dense network by lines, and those are family groups. I don't want to walk through this entire figure, but if we just focus on patterns of relatedness from the country of Guatemala, these blue areas on this map correspond to areas where we have characterized vivax malaria parasites from infected persons. And across the whole country of Guatemala, in most places, the parasite genetic profile is very highly related. So Guatemala on this network over here on the left is indicated by a purple color. And you can see two different purple balls, basically, that represent the clones that are unique to Guatemala that we've seen nowhere else. But in one location in Guatemala, Chiquimula, we see a more complicated pattern. And so I'm going to zoom in here and show you a close-up of this particular family group, where most of those dots are yellow, indicating those are samples that we collected in Panama, or that our collaborators collected in Panama. But you see a few purple dots. And so this is an indication, kind of like the story of my maternal heritage, of parasites moving from Panama to Guatemala at some point in recent history. And if we look at another family cluster that shares mixed colors, different color dots here. This one here has 3 different color dots. There's a lot of yellow dots, but there's 2 blue dots, which means that those are parasites originally observed on the Pacific coast of Colombia. So the connectivity between the blue and yellow means we think that vivax parasites made their way from the Pacific coast of Colombia to Panama, and then subsequently made their way from Panama to Guatemala. And so we can reconstruct in detail the way that parasites are moving around this region and understand if they're contributing to changes in incidence. And even though we do see some of these fascinating examples of parasite movement, if we look overall at the rates of parasites that look like they are indigenous strains or imported recently from a different country, the majority of vivax parasites that we see post-2020, when migration started to go up, still look indigenous in Panama and Guatemala. And so we don't think migration is directly responsible for the 4,000% increase in cases in Panama since 2020. So what could explain the change in cases if it's not a massive influx of South American parasites into Mesoamerica? Caroline Bucky's group has been looking at mobility data the old-fashioned way, by just asking people where they go within Panama. And one interesting thing that they have found is that in addition to the increased flux of people coming into the country and moving through it as part of larger scale migration, mobility within the country has also really changed in concert with this huge surge in migration. So on this plot, the gray bars represent migrants entering Panama, but the gray line and the green line represent movement of people within Panama. And the higher the lines go, it means the more geographic areas within the country is that they are accessing. And so we think that migration may have indirectly affected malaria by changing patterns of mobility within the country and increasing the interface between medically served and underserved populations. And credit to Caroline's group for this work. So to wrap up, mobility is important for understanding malaria, and hopefully the lessons that we learn in places like the Americas can be applicable to higher burden areas like Africa as they approach elimination. And that it's not always a simple story, however, of just migration resulting in the importation of parasite strains that weren't formerly native to an area. And so understanding the mechanism by which mobility influences malaria is important for informing the nature of the control actions that are taken against the disease. And then finally, as we talked about earlier, malaria, the Americas was the last region to be invaded by malaria, thank you to colonialism. And there is a prospect that it's one of the first areas where it's eliminated, hopefully with knowledge that will be applicable to many other parts of the world. So thank you.
Thank you so much, Dan. And now the next speaker is our keynote speaker for the day, Dr. Fitsum Tadesse. It's a real pleasure to have Fitsum here with us today. We're very lucky. Fitsum is one of the leaders at the Malaria and Neglected Tropical Diseases department at the Armer Hansen Research Institute in Addis Ababa in Ethiopia, and he's leading a very large initiative in the Horn of Africa aimed at malaria surveillance for both Plasmodium falciparum and vivax, and also understanding the role of invasive species in transmission and the role of failure of our diagnostic tests. So, Fitsum, thank you very much for coming. It's a real pleasure to have you here.
Thank you, Flamina, for the introduction, and thank you for the organizers for putting this together and for having me here. And what I'll be doing here today is I have an easy job. Already I have the scene set by Dyann, mentioning all the biological traits and everything, and Marcia mentioning the effect of climate change, and I'll be touching it a little bit, even if my focus will be on the biology, and then giving all the basics about genomics, the IBD, and all metrics. So I'm based at the Armor Hansen Research Institute in Addis, but also have affiliation with London School of Hygiene and Tropical Medicine. So I'll be giving you an overview of some findings we've been working on through Horn of Africa wide project, which is called HAMS, supported by the Gates Foundation. Okay, so a little bit of introduction. I'll start with Ethiopia, just like a snapshot from the region. It was having one of the most successful malaria control history globally. This is from WHO 2020 report. And all countries have been like having stall in progress or reversal, but Ethiopia was one of the 4 countries which were on track. And then it achieved the 2020 GTS goals of reducing malaria by 40% compared to its baseline in 2015. And this was linked with a widespread introduction of early lines, RDT, ACTs, and also more than 30,000 health extension workers being deployed across the country. And then because of this success, the program even has changed its name to Malaria Elimination Program, and then elimination efforts were initiated in 222 districts where the case burden has really went down. But this hasn't continued for long, unfortunately. Towards the end of 2020, we started to see a resurgence, and within only 5 years' time, between 2019 and 2024, we have seen a tenfold increase in case, from less than 1 million case, 900,000 case, to 9.7 million case within 5 years' time. So you can see how vulnerable this is. And because of that, we joined the party. And then you can see how much Nigeria is contributing. I always get surprised. Even if my focus is Ethiopia, I always mention this. It's massive, right? 26% of the global burden. And then now we have become contributing 4% of the global burden in 2024. Just pause there, and I'll tell you what has happened after that. So the resurgence has happened throughout the country, but it's mainly affected the western and northern, western and southwestern parts of the country. And the resurgence was not only of Plasmodium falciparum, but it also has resulted in a huge resurgence in Plasmodium vivax case, but the numbers were higher for Plasmodium falciparum. And in Ethiopia, altitude is an important factor because it shapes Plasmodium species distribution and composition. And when you look at those resurgence years and divide the size based on their altitude, the colors in here, you can clearly see that the lowlands, the faint colors, the lowlands have seen less increase in case because they were already high malaria burden for falciparum, mainly falciparum. And the midlands, the ones between 1,000 and 2,000 meters above sea level, have seen a major resurgence. And the highlands have seen vivax resurgence. And when you put this together, you can see that the gap between the contribution of the two is closing, but is at the highlands. So vivax is really increasing. And this varies between size. So the blue ones are where the proportional increase is higher for vivax, and the red ones are where the proportional increase is higher for falciparum. And then we try to look into available data to investigate what has caused this resurgence. And the first was on ecological drivers and human mobility. Unfortunately, during this same time when the resurgence has happened, the country has also passed through crisis, conflict within the country, and also the neighboring area. The Horn of Africa is a chaotic place. So we have seen that a lot of ecological drivers have been contributing. Enhanced vegetation index, increase in moisture, maximum daytime temperature, lagged rainfall, and displacement. But all of these factors combined, they fail to explain the entire increase in malaria case. So only 18% of the falciparum rise is explained by the combined effect of all of this. So what could have caused the remaining resurgence? So that was the study I've been leading. And if you look into WHO and other reports and recent investigations, you can see that there are a lot of biological threats emerging and converging. And these are the major 4: insecticide resistance, anti-malaria drug resistance, HRP2 deletion causing diagnostics challenge, and invasive vectors species. So WHO has them on its malaria trace map. When you look at their report, Anopheles stephensi was first reported in Djibouti and then Ethiopia and spreading westward. The latest report has it in Niger. And artemisinin partial resistance, first in Uganda and Rwanda, and then now it starts spreading in the Horn of Africa region. And HRP23 gene deletion that affects diagnosis. In Eritrea, before they changed their policy, there was a place which had like 80% deletion. Djibouti had 86.5% parasites being missed by the RDT caused by deletion. So this needed a coordinated effort in the region. That's why we have come up with this project called HAMS, Horn of Africa Malaria Molecular Surveillance, so supported by the Gates Foundation. And these are some of our study sites. So we also have Yemen across the Gulf. So I'll tell you a little bit of some of the outcomes from this study. So about the RDT issue. So RDTs have played a major role in improving diagnosis. Dan already mentioned this. And you can see the pink. They are pink, right? The color. Okay. Okay. So RGT supported like scaling up of diagnosis. Okay. So there are several antigens that we use for on RGTs, and among them, HRP2 is very falciparum specific, abundantly expressed. So it was the most commonly used RDT to diagnose falciparum malaria. But recently, we have started to see that parasites, which are detectable, which used to be detected by RDTs, are becoming undetectable, mainly linked with deletion of the HRP2 and also a sister gene, HRP3. And the first report came from Peru, and it was really a lot of deletion there. But what's fascinating there is Peru hasn't used RDTs, and the selection because of RDTs doesn't explain it. So reports from the neighboring countries, Eritrea and Djibouti, and the switch in change also motivated us to do studies in Ethiopia. So what you see in here, the domains colored based on their discordance, microscopy and RDT discordance, HRP2 based RDT discordance, and the pie charts indicating the deletion prevalence. So you can see that there is heterogeneity at country level. But when you look deeper into those specific study sites where the discordance and deletion is higher, you can see that there is at a micro level, there's a huge an incredible amount of heterogeneity within the study sites, which are really very close to each other. And this is also reflected on the HRP23 gene deletion prevalence. So these are 10 study sites per each one of the domains. So 100 health facilities were included in this study from the 10 domains. So using this data, we wrote policy brief document, and then the government already endorsed switch to non-HRP2 based RDTs. And we expanded the study recently to Sudan, and we have done surveillance around 4 states in Sudan. And the findings were very similar. So the borders near Ethiopia were the highest affected, and also the states near Eritrea were the most affected. And using the same data, which also demonstrated huge heterogeneity at a smaller geographical scale, we informed policy change, and it was a success for the program we established there. So overall, in the Horn of Africa region, the HRP23 gene deletion is massively spreading. We need some data to use the word spreading, but it's a big challenge there. Okay, so the next problem in the region we have been dealing with is drug resistance. So Plasmodium falciparum has developed drug resistance to almost all anti-malarias we have used. This is from a beautiful review paper from David Fiddock's team, and you can see that all anti-malarias, falciparum has developed a resistance to almost all anti-malarias. Artemisinin resistance has emerged in the Southeast Asia, Thailand, Cambodia border in the 2010, and then the recent reports from Africa are from Rwanda and Uganda. And there are different genotypes circulating in the region. There is a unique one circulating in the Horn of Africa region. It's called R622I on the Kelch gene. And the red colors are indicating the 622I, and the blue in here is indicating 675V, which was first reported in northern Uganda border. Northern Uganda is close here, is across the border next to South Sudan. So there's a heterogeneous distribution of the K13 mutations, a unique presence of R622I up to 50% of the parasites carrying it in the northwestern part of the country. But what's challenging in a setting like Ethiopia is we also have Plasmodium vivax, and we use chloroquine to treat Plasmodium vivax. And then because in co-endemic settings, patients who received previously chloroquine are walking around and getting exposed to falciparum, and then the falciparum parasites are still under continued drug pressure from chloroquine. So this is the CRT gene, the haplotype, CVIT, CVMNK, and you can clearly see that the distribution of the two haplotypes, the wild type CVMNK and the mutant CVIT, their distribution differs geographically. So if you put like a line there, you can clearly see that there is a divide. The major driver is vivax co-endemicity. So in the lowlands, where there is no vivax, there is less availability of chloroquine. And because of that, the wild type is reversed. But in the highlands, where there is still chloroquine because of vivax presence, the mutant haplotypes are still circulating. And then when you expand this to the rest of the region, in Sudan and Djibouti, you see the same thing. So R622I is circulating at a higher amount in the northwestern part of the country. But the moment we expanded the study to the southwestern part of the country, in the previous map, if you notice, there is no 622I that much. But in this study, that has happened in 2021, and this has happened in 2024. And one thing that has changed the profile in here is linked with the presentation from Dyann and Marcia. One of the study sites we included in this study has a gold mining area. And then we know that most of the workers there, they are seasonal migrant workers, and they often come from the northwestern part of the country. And some relatedness analysis, I'll show you later, has confirmed that the movement is contributing to this. So if you notice the distribution of the HRP23 gene deletion and the K30 mutation, it happens mostly in the northwestern part of the country. So there's ecological co-occurrence of the two. And then when you look into the details, individual parasites, there is co-occurrence of the two. So among the 622I parasites, more than 50% of them, 56%, they also carry deletion. So among the deleted parasites, almost 1/3 of them were also, the other way around. So the number is higher for the HRP2 deletion. So 1/3 of the double deleted parasites carry the K13 mutation. So we don't have explanation for this. We expect the other way around, right? Parasites escaping treatment because of misdiagnosis are less exposed to the drug. That's what we expect. But we see that the deletion, parasite escaping diagnosis, are also carrying the drug-resistant mutation. And then we're trying to understand this. So a little bit of the genomic background of the parasites circulating in the region. So this is, sorry, it's not very clear in here. It's all the three countries. The color codes outside are indicating the three countries, Djibouti, Sudan, and Ethiopia. And the colors inside are indicating the regions within each one of these three countries. I'll be bringing this later in detail. But the overall observation in this study is there is distinct regional genomic cluster. And within Ethiopia, we see that there are four major geographical clusters, semi-isolated populations: the central, the northwestern, the southern, southwestern, and the eastern parasites. In Djibouti, we see mostly monoclonal, highly related parasites, but they are strongly linked with Ethiopian parasites. So given this structured background, so our next question was, how do diagnostics, evasion, and drug resistance shape the structure of these parasites and also movement across the region? So this is, as Dyann mentioned it earlier, this an IBD network at a cutoff of 0.9. So clonal and near clonal parasites, and the shapes are identifying the K30 mutation, and colors are identifying the HRP23 gene deletion status. So you can see these clouds, same color and also shape, shared. So triangle plus a color is indicating a combination of the two. So this indicates that HRP23 gene deletion parasites are clustering tightly, and also K13 mutant parasites. So it hints toward this lineage-specific emergence and expansion of these parasites arising within a particular genomic background. And the co-occurrence of the two, hence maybe there is co-selection and under combined diagnostics and drug pressure, but that doesn't explain it, right? No. So we also see that the wild type parasites form expanding clusters. So like these ones. So there is a background effect, not only driven by the diagnosis and drug pressure. So when you look into them based on geographic occurrence within each one of the 3 countries in the region, you can clearly see that there are region-specific distinct dominant lineages happening, but some of them are also widespread, especially this is referring to the northwestern parasites, the ones that are carrying both the K30 mutation and the HRP23. This color, you see it's like distributed in most parts of the country. Maybe it's indicating it's a hotspot and from where all others are emerging and where they are expanding from, or a population thing happening there. So although we see that most of the expansion is local, subset of the parasites are also moving across borders within country and also across international borders. So this map is indicating K13 mutant and wild type parasites, and the red line is indicating the linkage between the mutant parasites. So this setting, the northwestern setting, seems to be the source population. Now we are looking into detailed genomics of these parasites, whole genome and also haplotype construction. So this data supports a model of transboundary movement and local amplification. So based on this data, we started looking into some whole genome sequencing. So we have done 120 whole genome sequencing of parasites selected from these 5 regions in the country based on the amplicon data I have showed you. And what you see in this PCA plot is parasites from Ethiopia, red, and parasites from Sudan in orange. So the parasites from the Horn of Africa, the moment you start to add, for example, Kenya, a neighboring country here, they are so distinct. And they are distinct in terms of their ancestral lineage and even compared to the ones near the region. And we try to compare them to parasites from across the Gulf, including Indian parasites, but we don't see that they are similar. So there is a unique parasite population spreading in that region. So the follow-up study we have done was we continued monitoring this over time. So annual sample collection and then monitoring if this is evolving and then what pace it has, and also working on predictive modeling to see if this is going to saturate at some point, and if there will be any intervention change, what will happen to this change in MFT, multiple first lines, combinations, a switch from ACTs, maybe other treatments. But that exercise is still happening. And then this is just a snapshot of the data. So we've been monitoring these parasites in different domains in the country. The color codes are indicating the domains. And the bar plots, the data points are indicating the years 2021 to 2024. So you can clearly see that in the northwestern part of the country, 622I is increasing year from year, and then the pace is really high. But what's fascinating there is the CRT gene. I just took it's a lot of things happening there. I just only picked CRT from the bulk data, so you can see that there's a unique phenomena happening there. The CRT, CVIT, and CVMNK, the wild type is increasing, while the K30 mutations are also increasing in that setting. So one thing the way we have observed was we have just completed a multi-site efficacy study in the country, and one of the study sites was here in Gondar. And all of them were included, all the 5 sites were included. And we have seen treatment efficacy ranging from 80% to 100% among the 5 sites. And one unique thing we have seen was delayed clearance was strongly linked with K30 mutation, but K30 mutation doesn't explain the treatment outcome. But when we looked into other genomic markers, trying to find any explanation there, we have seen that parasites reversing back to the CVMNK are most likely to fail the current treatment. but the explanations are not yet there, so it's still exploratory. So the other layer, as Flaminia mentioned, I'm not only working on the parasite, the human side of the parasite, but also working on the mosquito stage of the parasite. So we have this new mosquito, an urban mosquito, Anopheles stephensi. So we've been trying to see what would its contribution be in this region where all these challenges are converging. So the perfect storm. Dan used this term, if you remember. So Djibouti was almost eliminating malaria. In 2012, Djibouti has seen Anopheles stephensi, and cases have surged like this. There was no formal investigation of this. There was no any evidence. In a city very close to Djibouti, Redrawing in Ethiopia in 2022, during the dry months of the year, January, February, March, there was a huge resurgence of cases. And then I sent my team to do a case control study to investigate the cause of the resurgence. And the resurgence has happened in a university campus where students live within the campus and also within the city. And what you see in here is the distribution of the case and also the distribution of the Anopheles stephensi mosquitoes. So the reddish colors, the dark red, are indicating the overlap between the presence of Anopheles stephensi and the additional case we detected by following the case. So there was a clear evidence there. And then we have sequenced these parasites from the two sides, from the university campus and from the city. And what we have seen was there were 4 lineages. The 2 were the dominant ones. And the second lineage has happened mainly in the university campus. And the parasites detected in the university campus, they were all missed by the RDT. And they were confirmed to be because of HRP23 gene deletion. And they all carried the K30 mutation. So this is a perfect storm, right? So an invasive mosquito transmitting parasites missed by an RDT, and which also carry a marker of a validated drug resistance marker. So with this, I'll conclude my presentation. Sorry, I'm the sad storyteller in here. I don't have motivating news at the end of my presentation. So there is convergence of malaria biological traits in the Horn of Africa. And the HRP23 gene deletion and the K30 mutations arise within a specific genetic background, but there is evidence of local expansion and cross-border movement, and also within country movement. And there is also, maybe the motivating part of this is there is a good translation of evidence to policy within months and an opportunity to expand this to other regions. Maybe this is time we might have to start thinking about mitigation before it's too late. Maybe it's already too late. Thank you very much.
Thank you so much, Fitzum, for taking us through this incredible investigative work that you and your team are doing. I should also mention that Fitzum made the New York Times with his work a couple of times, but it's too modest to talk about it. But really, so please, yeah, Marcia and Dan already ready for the Q&A that we are all waiting for. And Frederick, please come back.
Thank you very much to our presenters. I think we can begin taking our questions, and this time I'll start from the back. Can you please use your microphone if it's working? No. Okay, maybe I'll get you an extra microphone.
No, I think this one not working. I'm coming here. Okay, nice talk, all of you. The first question goes to the first and last presenter because the climate change is that we have a lot of biological threats. So especially for the malaria resurgence we're seeing in Ethiopia plus Brazil presentation, how we can count the biological threat versus climate change. So is the climate change is really contributing or the biological threat is contributing for that one? And Danny, nice talk. And my question is, the movement of parasites, so the directionality, so is that going to one direction or coming back? How we count the directionality by IVD measurement? Thank you.
Thank you.
All right, thank you for your question. So, I think one way to think about this is this whole discussion about climate. Climate comes and exacerbates problems you already have. So it becomes another layer that makes inequalities worse. And if you think about the challenges and what Fitzum just said, it can make things worse. Now, which one happened first? That's the chicken and egg question, right? There are probably ways you can model and you can try to quantify the contribution of each one and the interaction, how much climate made something else worse. But I think the most important thing is there are things you can try to do in addressing the biological challenges. But for the climate, you need a lot of political will, you need a lot of action, you need to have early warning systems. Just the early warning system is not going to do anything, but you need protocols. If there is a drought coming or a flood coming or a heat wave coming, what should you do? And what are the roles and responsibilities for each one? We have very few places that have all of this detailed in place, and when things are about to happen, they actually do something. We can't avoid the climate change because we're really not doing things as we should. So we better have those things in place so we can not be reactive, but can anticipate those things and make them, less dramatic as they could be.
Following up on that, in some of the maps I presented in the beginning, there were like gray areas. And one of the reasons why those were gray is like the Tigray region, there was war, there was no data. Some of the parts of the country, the quality of the data was not something we can use in the model. And we also tried to include biological information together with the ecological variables. But one of the challenges we had was the drug resistance data. We just started generating it. HRP2 deletion, everything. It wasn't really covering a good proportion of the country, the Stephen site. It was very sparse. We included it in our model in the beginning, but it was really not a proper way of including it because the Stephen site data, like at the zonal level, which is a bigger geography, and then classifying that zone as Stephen site positive, it wasn't a good judgment because the country is so heterogeneous. I told you like altitude is really an important factor. Addis Ababa, for example, the capital, it's like 3,000 meters above sea level. There is no malaria if you go to Addis. But you drive 40 kilometers outside of Addis, there is malaria. So the data availability, quality, granularity, everything was really important.
And maybe to answer your question about how to infer directionality from relatedness data indicating mobility, this is a difficult problem. As you know, I think there are some good statistical approaches that people are thinking about for doing this in higher transmission settings. In lower transmission settings, at least in Mesoamerica, what we have seen is that foci of transmission become isolated and develop a local clone or a couple of clones that tend to be really specific to a geographic area. And so this simplifies the inference quite a lot. As long as one is able to work with a strong kind of baseline survey of what local diversity looks like. If you knew that 2 years ago or 5 years ago, this was the distribution of clones, and in a new sample, you see something from country A in country C for the first time, I think the inference of directionality is simpler. And hopefully as other regions approach similarly low transmission levels, these kinds of patterns will also be able to be inferred from genetic data.
All right, thank you. Any question? Okay, no.
All right, thank you very much for your presentations. quite remarkable. I like the phrase "the perfect storm." I think it's a huge problem we have. My question centers around both presenters, Marcia and then Fitsum. For Fitsum, I think you talked about uh, expansion of lineages based on migration patterns, and I think you talked about annual tracking. My question is there a basis for deciding on an annual approach? When you see that migration happens continuously evolving, I'm wondering if you want to vary between maybe manual, biannual or quarterly. Because I'm wondering if there are any variations, with respect to that and how that might influence your outcomes. My second question to Marcia is, I'm curious why you picked on the Amazon forest. I'm from Nigeria, and we also have, several deforestations happening and even drought. But I think that the Amazon forest is very peculiar. I mean, as an ecosystem, I'm wondering if you can actually translate or, extricate the findings there to, to actually apply to other settings that don't have the same peculiarities as the Amazon forest. Thank you.
That's a nice comment, but the answer is just about the resources. Yeah. Yeah. Even the annual study collection is a big country. It's 130 million people, and then the study sites are like 1,000 kilometers each other. And then driving there, collecting the samples, bringing them to the lab, processing. It's not an easy task. Yeah. This also depends on sustained funding, acquiring the funds. Yeah, but it's an interesting question. But there are efforts now happening on intervention in migrant populations using second-line treatment, DHA peppercorn in the country, and we're discussing how we can integrate this kind of evidence generation with those implementations.
So let's see, two things: why the Amazon and how this can apply to other settings. I'm from Brazil. I've been working in the Amazon since '99. 100% of my work is in Brazil. I'm committed to the country. I always study in public school, so that's my way to give something back. And it's an absolutely fascinating and challenging ecosystem to work in. A lot of what I showed you applies to other countries in the Americas that share portions of the Amazon. So we have collaborations with Peru. Some of the challenges are similar: the mobility, the deforestation, the mining, the challenge of riverine communities. Colombia also has similarities, although Colombia has malaria in the Amazon and in the Pacific, which is very different. Does the work in the Amazon apply to Nigeria? Probably not. But the approaches, the frameworks, they apply. And they have to be modified based on the contextual issues in Nigeria. So the way of thinking can be adapted to other areas. The findings, no. It's particular to that ecological setting of the rainforest that shares similar challenges across the region.
And maybe I can add just one brief comment from a collaboration that Marcia and I have had. In the Amazon, the most important mosquito vector of malaria, Anopheles darlingi, favors habitat at the edge of the forest where there is a clearing. And so forest clearance directly enriches the habitat of the most important vector of malaria. So the degree to which those observations translate to other forested settings, I think depends on the local ecology of those vectors. And in many settings in Africa, there's different primary vectors and long lists of secondary vectors that are going to affect epidemiology in the context of deforestation.
All right.
Can I just add one thing to that? Sorry. I think this really does emphasize this issue of subnational tailoring, that each district or each unit is going to need its own information. And I feel like this is like a revelation that we should be emphasizing. So, and I agree with you, the methodology can be the same, but the data is going to be different.
Okay. Thank you very much. So I'll take Deepal. Yeah, sure.
Well, okay, Diane's taught me well because I have a related question. So If we're thinking about sort of sub-country level tailoring, I'm curious, given that you've given all these examples of how geographic, like either climate change or economic things are moving populations, are there national malaria control programs that have built-in strategies now that seem like they're going to be able to do sort of regional tailoring, but with some flexibility to account for movement of where you have intensity? Or are there, if not, are there particular strategies that you think would be useful?
I mean, I can tell you what's happening in Brazil, and there's a major effort from PAHO. There's a new guideline that will come out soon with the strategies for 26 to 2030, and exactly that is right there. Either you really make changes to move towards elimination, or it's going to be like the cartoon from Einstein that Dyann said. You're doing the same thing and expecting different results, which obviously is not going to happen. So what the program in Brazil has been doing now, because there's this massive increase in indigenous and mining areas, is to deploy folks that stay in the capital working in the program locally, and basically design different things to do in each area. And this makes a heck of a difference, not only because you're tailoring, but because you're empowering those people that before had never seen somebody that works in the ministry, and they feel valued. Their knowledge is also informing, because there are things you only find out when you go there. And that's what I tried to mention at the end. You have to work with those people as well, because this kind of empowerment locally can be a huge thing, making a difference when you're moving towards elimination. So I am not going to claim that Brazil is already there, but those are good first steps to achieve what you said. The thing is, all of this is very fragile, because honestly, public health is political. I don't care what people tell you, it is. And we have elections this year in Brazil. And if we go back to what we were 4 years ago, that's going to be a mess. I have to say, the National Malaria Control Program was the resistance. That's why I was there. And some work will continue. However, the funding landscape can change. So we are in the good track, but depending on what happens in October in the elections, I hope this doesn't happen, but the resurgence, remember those graphs Dyann showed, you relax and then you go back to where you were.
Okay, so we'll take just one more question. I'll take it from the back, and then... No.
Thank you all very much for this. I know nothing about malaria, so this is very insightful for me. But I think it does touch on like general themes for all infectious diseases, right? Like migration is a big thing. Global warming is a huge thing. And I do feel like infectious diseases in general can, because it's a public health issue, can benefit a lot with, like Marcella was saying, like involving local people in sort of like crowdsourcing data for other folks. And so what is the state of point of care diagnostics in malaria? And is there an opportunity for these kinds of diagnostics to help collect information about whether the vector or the patient or the bug, basically, or the bacteria itself, to sort of inform very rapid interventions? So people collect samples, it gets shipped to a central lab, maybe somewhere in Addis Ababa, or and then you get this data in a very, very fast way, because it seems like the world is moving very fast, and this kind of like very rapid feedback of data and information to decision makers is critical, whatever the infectious disease is. And in the case of malaria, I just have no understanding of like where point of care diagnostic thinking is and what are the obstacles to rolling it out across different regions might be.
Okay, so we have the diagnostics, we have RDTs, we have microscopy, we have everything. But when you think about like a country which is dealing with 10 million cases annually, that question you raised might be not an easy thing to do, right? Yeah, the burden this might be different in a different context, in a low endemic weight of country.
Yeah, so every country is going to be different. That's the thing. You can take all malaria endemic countries and they're different. They have different health systems. They have different health information systems. They have different networks of labs and facilities where people can get tested. So I mentioned when I presented Brazil is unique in the amount of data. So there's an entire form that collects information for every case. And the key question is, you can collect the best information possible, but if you're not using this to create evidence and then take action, it doesn't matter. And that's what we're trying to do now, is to leverage this sheer amount of data that are just sitting there, that and don't just make a dashboard. I'm so tired of dashboards. But if you really use this to inform what should be done, the subnational tailor, what you should do in each one of those small areas, not in the entire country, that's the sort of leapfrog that even countries that have data, they're not there yet. But we have to do this, because if you don't know, you don't resolve.
All right. Thank you very much to our presenters. We will end this question and answer session here, and then the next set will continue. Thank you.
And we have now a 20-minute break. Actually, no, 15-minute break.